Good morning, everyone. My name is Lynn Bree. I'm director of the Massachusetts Host Microbiome, pathologist at Brigham and Women's Hospital. And I'm delighted to introduce Dr. Wenyuan Shi from the Forsyth Institute, who is going to speak to us about structure, function, and manipulation of the oral microbiome. When asked how he got into microbiology, he requested that we take this opportunity to honor his thesis advisor, Dr. Julius Adler at the University of Wisconsin, who passed away a couple of weeks ago. Julius pioneered our understanding of methyl acceptor chemotaxis proteins. And I want to quote Yuan Wen's specific words because I thought they were very insightful. He said, "My childhood dream when I was growing up in China was to become a fortune teller. Then I learned that Americans had created a new science called behavioral genetics. I thought that this was cool since it is a scientific way to do fortune telling. That is how I chose genetics as my major. And I did my PhD with Dr. Julius Adler on E. coli chemotaxis, which led me to become a microbiologist." So Wenyuan, we wish you continued good fortune and are looking forward to what you're going to tell us.
Yes, I'm glad that the chemotaxis have been mentioned, and Julie says the founding father of the chemotaxis. It's so sad that he passed away, but he lived a great life. And I just very fortunate 40 years ago when I walked into his lab and he talked me into making my PhD thesis on trained E. coli to learn. And that definitely, putting me in a different perspective when it comes to microbiology. So thanks for the committee and to give the opportunity. Lynn, thanks for the great introduction. So I would like to start with introducing Forsyth, and we're not very far away and have been around for 114 years. This is one of the premier, should be the best, oral health research institute. But if you're ever wondering how dentistry have anything to do with microbiology, that was the very first microbiology paper human ever published. And if you're actually reading that paper published by Leeuwenhoek back in 1677, and which he made the discovery in 1676, and he somehow figured, oral cavity must have a lot of bacteria. He decided not brushing his teeth for a couple of days, and then using that and put it under the most primitive microscope, and that's what he see. That was the figure 3 on his paper, and now it was very interesting. The next 300 years, we've been all doing this one bacteria, one disease thing. But the very first microbiology paper is already showing you the complexity of the microbiome in oral cavity. There's many different kinds of bacteria. They were all together under one primitive microscope field. Now, 350 years later, and we know a lot more about those bacteria, and which I will show you some of the actual figure. But those little dots, the figure E, are the streptococci. The oral cavity have over 50 different kind of streptococci. Most of them are commensal, but limited few, and especially after human learns to make a sugar out of sugar canes, and we're eating so much sugar and changing the oral ecology. That's why the toothpick decay becomes so epidemic. And a lot of other gram-negative bacteria you can find in the subgingival who directly responsible for the periodontal disease. But it's even more exciting recently, if you see some of the recent Nature paper, everybody talking about how the fusel, which is the oral bacteria, travel all the way to a colon and responsible for the metastasis of the colon cancer. And that was actually the very first bacteria and Leeuwenhoek observed back in 1676. And the spirochetes, you probably also heard the deep connection between oral cavity and Alzheimer's disease, and the more bacteria from the brain banks, you can actually, most frequent bacteria can detect it in the brain. Alzheimer's brains are the oral bacteria. So this is also suggesting a deep connection between oral cavity and the systemic disease. Now, all that discovery, I have to be very proud to say, and they all have something to do with foresight. And when you look at, in the way I think it's very exciting being a microbiologist, the last 40 years is just so much of the changes in the entire microbiology from, the concept of the biofilm, by Bill Costigan, to the amazing advance of the sequence technology and to the concept of the ecology and look at the whole community, to HMP, to National Microbiome Initiative. And the one thing I want to be very proud to say is oral microbiology goes through the process. Not only they go through the same process, many cases we're actually promoting and trigger this and really in the leading field, such as the microbial ecology concept in disease. And the idea was actually developed in 1979 and by the oral microbiologist. And if you look at the NIH, go to the Congress in the 2006 for starting the second human genome project of HMP. They're actually using the data out of foresight as a justification to say, now you need to do the same thing on the skins, vaginal, and the gut. And it was just such an amazing story. The history in foresight is along the way, and we have so many of the amazing scientists have been contributed to that. And I'm also very happy to report, and 5 months ago, and we now integrated with the American Dental Association and the ADA, and have been oversee the 79 dental schools, education accreditations. They are providing the clinical guidance for the entire 250,000 dentists. They have direct connections with the congressional leaders to determine the health policy. Now we be able to adding our innovation into that part of the process. So this is very exciting. I was using the title when we announced the merger. Boston Globe used the article that the biggest monster, is created. And if ADA was a 800-pound gorilla, and we're trying to make it a 8,000-pound King Kong. And so for that reason, and I'm here, wanted to give you some sense and give you some overview about some of the great work we do on the science, but I also want to take the opportunity inviting for the collaborations, because now those days, this time is right, the connection between oral and the systemic disease. And oral with the environmental microbiology, which I'm going to get to it. There are just so many, collaboration opportunity we have. So I want to thank for Michael and Peter and all that opportunity to showcase what we hear. But before I, get to the actual science, I want to say I joined Foresight 7 years ago, and it was just such an amazing experience. Have the opportunity to work with some of the amazing microbiologists, which I want to acknowledge them first to get started. Okay, so when it comes to the structure, and Foresight did every other microbiome group people is doing, and you are sequencing it, you understand what they have, and you can actually that's about, 500 to 1,000 species. It depends on who's going to doing it in the gut. But oral bacteria actually have as many and as in the gut, but have a lot of uniqueness. And you can see the first 12 or so is dominating the majority part, but you really have a very long tail. And it's a true jungle. Not only you have the eubacteria, but also you can find Archaea and all those there. Now, one thing we're very proud and led by the Dr. Floyd Dewhurst is unlike other microbiome, and Alex just mentioned less than 1% of them are ever being characterized. We actually characterized and cultured 70% of them, and that being able to culture them, we can do so much biology. We can really understand it, and which is truly cool. I want to share one first story is. We probably have the best bacterial culture laboratories in the world, and we have all kinds of crazy medium and manage to culture so many of those bacteria. But yet, we not be able to culture the 30% of those. And now we start to know why. And So if you look at the tree of the life, and Joe Banfield's lab, is give us some new perspective. And you see their CPR, and they're actually 15 to 20% of the biomass. But you can, using your sequence method, you can find them everywhere, the ocean, the earth, and the human body. And so assuming they can grow anywhere, they must be very easy to grow. But yet, no one ever be able to culture them. So many of the lab was tried. Now we can actually find a lot of those among the 30% of the unculturable bacteria in oral cavity. So we actually, my lab took this approach, which could be used by a lot of other people. And what we did is, you look at the middle of the saliva, and this is a DGG, each band showing one of the bacteria species. And original saliva have a lot of sample. Then you take all the commercial culture medium, and then you're tracking them. Each of them only be able to support a very small fraction of them. But you can see, because they're supporting a different fraction of them, so if you can combine some of those together, what I call the nutritional mapping, and then you combine together, you can create a medium. They actually name after me now. It's called Shi culture medium, which is actually commercially available. And then you can see you could actually, to getting most of the oral bacteria cultured at the community base. And so when you look at that, you suddenly notice the TM7, which is one of the CPR, very famous CPR organism, is suddenly in there. And now, since we've been growing them leaders and leaders, so allow us to concentrating them, yet we never be able to culture them. And until we had this aha moment, because every time when we do a single colony and try to sequence and see what those bacteria are, and we're getting this fraction of them always give you around sequencing data. And you always think, that was the bad firm you go to, or vendor. But when we try enough of the vendor, we see something is wrong. When you look at the microscope, that's the aha moment. Now, I'm very excited. It's actually, those bacteria figured that out a long time ago in the deep ocean, and they just used the same mechanism and come to the oral cavity. So the red is a host eubacteria, is actinomyces, but it was surrounded by those green dots. So if you're taking those green dots and then you sequence them, that's where, this was the TM7X was the first CPR organism ever cultured. And now there's so many people following the same way we do, and there's so many CPR organisms now being cultured. But when you're taking that and analyzing it, and it's exactly like in the deep oceans or the deep underground, and they have such a small genome, and a couple hundred thousand base pairs, and such shrinking genome doesn't have any essential amino acids and other things. They really rely on sucking the nutrition out of their host to survive. But that parasitic life, which is we now actually can culture the pair, and have been allow us to understand so much more about how bacteria interacting with each other, which I'll get to a little bit on the functional part. So thanks for that kind of approach. That number down to 25%, and about yet to figure out what do those other ones, we're going to figure out how they do it. But what Forsyth is really trying to do is not just limiting the sequencing and who they are. And thanks for Gary and Jessica, who is at the audience. We really try to understand the spatial structures, because every time you're sending them through the sequencing machine, you completely lost the spatial information, which is critical. But how you be able to actually understand them at the spatial level, where they are, and it has become something critical important. And thanks for the visionary works of Gary and Jessica, they developed the spectral imaging system, which I don't have the time to get into the detail, but they can really pretty much look at every analyte within the microbial structure. So now we know, and they're unlike a gut, it's like a big fermentation tank. They're all over the place. Oral cavity, And they get, ever since they matured at a year 2, they with you for the rest of your life. And so it's so well organized structure and make it so interesting. Now, we got this art science thing. I'll give you a candidate for next year. And Tavita and in the Gary and Jessica's lab, they now figure out so many good ways and to make that resolution become stronger, better than ever. There's so many other ways. I just don't have the time to showing all the slides. But this is just give you the flavor, and I'm sure Leeuwenhoek will be very happy. And 350 years later, we can take his original discovery and to find an amazing brand new world. And we actually pushed that step further. And in Jessica and Gary's lab, and they took the HMP, the Human Microbiome Project, which they took 9 different sites in the oral cavity. But the sequence they published is only at the genus level. So you can't really tell if there's any difference between those different sites of the body. But they're using this oligotyping method and to get the resolution to the species level. And then the magic happens. You notice, and there are only, this long line with all the species, and you can see, and certain species is only showed up and on particular location, even though they are all in the oral cavity. The bacteria in the tongue dorsal, and in plaque, and mucosa are totally different. And it's a different, I mentioned there's 50 different kind of streptococci, and they're actually spread in a different place almost exclusively. And there are different rossia, and they were actually one in tongue, one in plaque. And so this is really give you a new resolution to understand that there's a lot of site-specific microbiome, and within that oral microbiome, it's not a one microbiome, it's like even you're growing a culture in the single bacteria. And from Kim Lewis' work, we know there's a persister, there's other things, there's a huge diversity there. And this is to give you the flavor of how complex the community, who is already very complex, but this is a spatial microbiome is adding you the additional complex level. And so you make sure you study the right bacteria at the right place. And Gary and Jessica lab are pushing their science even further. They develop in the analytic tool they call the meta-pan genomics. And not only you look at the individual species, whether they are site-specific, you could actually look at the genomic information, finding whether you have any specific gene and who are responsible for that specific location, and they are. And we yet to figure out why that specific gene region and are helping the bacteria to the tongue or to the plaque. And I would love to someday doing the experiment, switch them and see whether we can actually change the location of the bacteria and go to the different place. Just like, we know a lot of the protein have leading sequence and sending them to the specific location. Looks like within all the species in the microbiome, everybody carry their community IDs, their zip code and area code, and they have to rely on that and to finding their friends and foes and go to the right community. And without the car key, you can get this building just like that. Okay, so I give you some flavor about all the works we're doing on the structurals and most of the microbiome work and stopped at the sequencing level, but there's just so much you can do. And through the culture, you can understand the biology better. And through a lot of those detailed spatial microbiome analysis, and you can get so much deeper information to understand the biology. Now, the other things I should be very proud to say, and what makes Forsyth is very different from a lot of other microbiome research groups is with so much emphasis on biology. And we've been using so much of the bacteria genetics, physiology to understand what's behind all the sequencing data and all those spatial data. So I'm going to give you a few examples for you to get the flavor of about the interspecies interaction and and inter-microbiome interaction. In a second, you know what I meant. I would say the ultimate frontier is the interaction between the microbiome, our host. And being a microbiologist, we would like to think it's the microbe who are trained our immune system. But actually, in many cases, the foresight study have been finding it's the immune system who determine what kind of bacteria there and what issues they are. And this is a very deep topic beyond me, but Dr. Tim Van Dyke is one of the few immunologists in the audience, and find him and chat a lot more. He has some very fancy story to tell you their interaction. And then lastly, I'm going to quickly cover something, Roberto. I'm actually attributed that to you. I'm going to get to that, but I'm started with the interspecies within the same microbiome, since trained by Julie's. And now I said I'm going to level up. Julie's focused on single cell and how they sensing the signal. And we really now see the microbiome is a bigger family there. So I telling the people we're studying bacterial politics, and which they need to have a good reason for politics, because every niche environment they stay, there's only limited food and nutrition in the space. So they have to fight with each other. But at the same time, they need to maintain the diversity, and they need to have a good interspecies interaction. And in the case of TM7, if you're parasitic, and then you want to suck in the nutrition from your host, but you don't want it to kill your host, because then you have nothing to suck on. And so this sophisticated competition and the cooperation balance is become one of the most fascinating things. And there's so many labs have been working on it at Forsyth with a very different story. I only have time to just give you a limited few. And one of them is certainly because we'll be able to culture the TM7 versus its host, we'll be able to do so many biology, and it was just amazing and fascinating. And now we're developing the genetics on the both ends. So we actually know what takes for the parasite. The host don't want to get rid of the parasite, and what it takes for parasite and to go there. And one of the things is very interesting is they're very host specific. And each of the TM7 only find their own host. But at the same time, they being the parasitic one, you need to be quickly evolved just like a virus. So actually in the oral cavity, it's not finding one TM7. You have 6 different groups, many different species. And it was in the very short order of the human evolution. And some of the evolution biologists tell me it was less than 100,000 years. And there's 3 different independent events. 3 different TM7 jumped into human oral cavity alone. And so this is, and they have a lot of ability to keep changing their morphology and the interacting partner. And you know why I have question of 11 and 1, and because in this case, they really want to amplify as much as they could, but they don't also want to overburden their host to kill them either. And so it was a fascinating biology, and we have a lot of paper in this area, and I don't have the time to go through detail, but I just want to give you a flavor of it. My favorite story happens in my lab. Remember Leeuwenhoek find the spirochetes and the fusel. There's a disease in dentistry called fusosporosis. And if you're getting a spirochete and the fusel at the same time, it could just destroy your gum in 2 weeks, which is normally takes 60 years before you're getting a periodontal disease. Now, why those 2 bacteria gets together and it becomes so deadly? And we've been looking into deeper, and to make the long story short, is a fusel and have the ability to trigger the cell deaths, and they're pretty deadly. And however, they're not very invasive. And so therefore, and the fusel will mostly do a damage on the surface, they're not getting deeper into the cell. The only reason they get into the colon most likely is the immune cell eat them, macrophage, and they move around and they deliver the fusel to somewhere else. Now, spirochete, on the other hand, they are move around in the body. They have using their chemotaxis mechanism to looking for the weak spot. And I was showing the bottom here, this blue with the dead cell. You can see the Y-type spirochete, they go nowhere but where the dead cells are. But if you have a chemotaxis minus mutant, then the spirochete is spread all over the place. And so the part of the reason they're getting together so deadly, as you can see here, they actually can bind together. And because of this binding, and so even though spirochete is very invasive, but they're not very pathogenic. And while the fusel is very pathogenic, but they're not very invasive. But they figure out a way to bind together and piggyback. And so spirochetes actually carry the fusel to the deep layer. And in that way, they causing a very acute tissue destruction and necrosis. Okay, so I didn't design this experiment by design, but actually what you talk about is make this perfect. And now we're talking about E. coli. And so one of the things is, this is where I'm going to the microbiome interaction. So one of the things is very interesting when you look at the HMP. And even though you got 1,000 bacteria, 700 here, maybe another 800 here, they hardly overlap, even though you swallow saliva all the time. And most of the oral bacteria have to go from here to get there. But they don't stay here. They actually just decide to go to the gut. And some other things Michael and I were talking about, this head and neck is one confluent microbiome and immunology. But when you look at the nasal, eye, and oral may be the reservoir, but their microbiome is very different. So there are some, especially happens on the borderline and what they decide where to go. And I'm sure the environmental microbiology have the same issue, the different layer, you got the different microbiome. And what determined that is become something we become very interested. And this is, goes to Ashley's finding, is E. coli, it's just so easy to get to your gut. So this is the experiment what we did for fun, is you taking all the bacteria from saliva, and which we call the omics, then you take all the bacteria together and with Shi medium and with the GI mix. And when you're creating those community, and then you take in isolates from oral or gut and throw them into the community. In this case, we did the E. coli. As you can see, it's no problem for E. coli to be easily become part of the GI community. That's why I guess it's so easy to get there. And however, and if you throw the same E. coli to an oral community, and in a few days, they were all gone. And in mice, they're eating their feces, which is full of E. coli. But any healthy mice, you do their oral microbiome, you couldn't detect a single E. coli. And that means they don't stay there. And why is that? And now, Roberto, I look forward to your talk, and I love the statement, future is in the past. And to people old enough like me, we know the Kohala library. I don't know how many youngsters ever heard of this. But that was the E. coli mutant library, and for you to initially getting an idea. Because we're finding when you throw the E. coli into the oral community, they will be dead in no time. So we decided to go through the Kohala library looking for E. coli mutant and who will be survived. So we can get some molecular mechanism and why E. coli only go to the gut, not in the oral. And what we're finding is certain mutants have a much reduced viability, and all those are involving oxidative stress. So which is suggesting the oral microbiome and is creating the oxidative stress to kill the E. coli. But that WAAL has got me. I have to go back to my old biochemistry book to figure out what's going on. What is that? Why that mutation could actually make them survive better? And it turns out that's a sugar transferase who are adding the enteric antigen on the surface of the E. coli. So with that mutant, the E. coli become naked. And oral community no longer think it's enteric bacteria. So they leave it alone and don't kill him. And so as you can see the study, E. coli alone doesn't, so this blue assay is to showing the production of the hydrogen peroxide and the oxidative stress. And you can see E. coli alone doesn't do anything. All mix have the capability to do it, but without any stimulus, they don't generate hydrogen peroxide. And if you're adding an oral bacteria like actinomyces, they hardly have any reaction. But the moment you add E. coli, they suddenly generating a huge amount of the oxygen. And that is based on the recognition of the enteric antigen. And so, fascinating. And this is really a good time being the microbiologist to look at the molecular mechanism and how the bacteria using a various of the tools and for them to do that. So that is ask us to ask an even bigger question. We already talked about they have a very well organized structure and they do a lot of metabolism cooperation. And given their social organism and all the other social organisms have, king and queen. And so it was one of the fun questions in my lab is just like in Julie's lab. He wants every graduate student come and to study the E. coli learning project. So I would have every graduate student postdoc come to the lab, and I've been always talking to them saying, hey, let's just think of what qualify a bacteria to be king and queen, and figure out some experiment to do it for me. And Roberto, you have always been my inspiration, and I was telling my graduate student and say, reading Roberto's papers and find some inspiration. And one of them did. And you still remember that old experiment you did with Steve, and you're taking the E. coli culture and seal it and throw the keys away. And after many, many years, they will still have some E. coli there. They're living on the dead body of their fellow E. coli. So we decided to do the experiment. What happened if I take the whole community of 700 species together? Then we throw the key away. And what happened? Let them compete and fight, and who will be the ultimate winner? The concept is we may not find the king and the queen, but we're at least going to find the cockroach and a rat, The concept is if you seal this place, let's say North Korean, King is not going to be the first one to die. Kim is not to be the first one to die. So that was the concept of the experiment. And it was a pretty fun results. And so Roberto enjoyed this. And first of all, just like E. coli, when you're counting the total CFU, and it's reduced, but to a certain level, they reach a plateau. You always have certain bacteria live there. And but when you can look at plating them, then you can see you started with all kinds of species there, but then later on, they become a very limited few. And then when you sequence them or RNA-seq them, and what you are finding is some of the most popular population initially to get started, like a Vonella and the strep. They are the ones that had a very small genome. They grow faster and they be most dominant. Suddenly they have no nutrition. They are the first one to die. But the two who managed to survive are the Klebsiella and the Providencia. And we're yet to get into the detail of the mechanism. The data suggesting is when you seal it, then you no longer have the nutrition. Those are the two have the biggest genome. And so they be able to actually synthesize a lot of essential amino acids and other things. And that's part of the reason they managed to live longer. Okay, so I'm quickly go to my last topic. I'm sorry, I will try to use limited 30 minutes to cover as much as we could. And the manipulation part. And so one of the goals we really try to do is to changing the surgical model into a medical model in dentistry, as you remember right now, and we were all doing that. So we being in order to do that, we need to do early diagnostics and the therapeutics. So first, I've been developing those old tools of doing the diagnostics and therapeutics. None of them are being, they all have their defects, either a low sensitivity or you have to have a lab to do it. And, but the CRISPR technology and out of the Broad Institute and creating this very unique opportunity. And because when you use like a Cas13 or Cas11, you can amplify the DNA and the RNA unlimited with a very simple device. So we've been now very effectively through a strategic collaboration with Sherlock Bioscience and developing that tool, and we're going to be very effectively making that on that. And lastly, to wrap up, is in addition to the diagnostics, we needed to effectively treating them. The biggest problem with the microbiome, the multi-species, when you use a broad spectrum killer or mechanical removal, you leave the space wide open. It's equal competition to go back. You always end up with more pathogens. And so we'll try to do something better. I'll just do one quick story is American Indian have the worst tooth decay and 97% of this, disease penetration. We asked the question, what happened to the 3%? And when we analyze the 3%, as you notice in the blue bar, those are the ones suddenly have this tongue bacteria jumping into the plaque. And that bacteria actually have the ability to stop the acid production by the strep mutant. So that is become a great probiotic reagent. And learn from that, we also have a lot of chemists at the Forsyth and to developing a pH, the pH-sensitive molecule. So their activity and is based on when you have a lower pH, they become antimicrobial. And at the higher pH, they aggregate together, lost the activity. So with that kind of way, we can treat the community, only get rid of the acid producer. So this is one of the few of those ecology-based antimicrobial treatment and showing very effective. And my last two slides is this one cool technology we've been developing and what we call the stamps. So essentially the concept is we're taking a broad spectrum bond and adding a homing device in front of it and allow them to do a targeted delivery. And luckily, bacteria doing all kinds of signal transduction. There's a lot of those signaling peptides and they're species specific. So we've been developing one of those and called C16G2 and showing a very specific killing. And we thought that would be a very good tool. Not only is showing the clinical efficacy, it's being developed as a drug. This is also going to become a very strong scientific tool. If you want to study microbiome and what each of the species are within that, you need to, just like the gene knockout, you need to knock out individual species within the mixture. And or you'll be able to isolate them and putting them back to the community. It's like overexpression versus gene knockout. So our species-specific killing is actually providing the unique opportunity, allow you to actually experimentally verify who are the keystone species. With this, I will thank you all for listening and happy to entertain any questions.
Thank you, Wen. That was an amazing trip through the oral microbiome and beyond. I have a question of the use, and I address this to everyone, and especially medical micro, about, in particular, say, TM7 being a parasite. Mm-hmm. What about if perhaps, it seems like with such a reduced genome and such a finite specificity for host, that potentially in the environment in the lab, they're kind of out of kilter, but potentially in the oral microbiome or wherever these pairs live.
Yeah.
And I'd say the same to you.
Yeah.
Maybe there's...
So because we be able to culture them, I know what your question are. We actually know a lot of mechanism. TM7 makes sure the host didn't get rid of them. So if they're actually making a metabolic contribution to the host, there are some specific arginine that's probably responsible for why they find the right host, who happen to know their expertise, they can contribute. Then the other, they have a lot of toxin, antitoxin tool. When you get rid of me, you'll be dead. So there's a lot of interesting way and for them to balance out.
So I would view that more as an evolved mutualism.
Yeah, eventually it could be the mutualism, yeah.
Thanks for that talk. Anytime I've seen a dense microbial ecosystem, there's a lot going on that's metabolic. How do they cross-feed? How do they compete for nutrients? So I'm just curious what you've learned to get ecosystems that close, even though they're structured and that dense. What are the metabolic interactions and what opportunities do they provide to enhance, disrupt, and potentially impact patient care?
Yeah. So the way I see it is, besides the host pathogen interaction, understand that at the metabolome level of how microbiome interacting with each other is super important. One of the things we already know is that so many bacteria generating their own antimicrobial compounds do their chemical warfare. Whether it's a production of the hydrogen peroxide, they kill E. coli, or a lot of those bacteria generating bactericin, who are extremely well regulated. They only spend energy generating bactericin when they need to. But certainly something even more. And one of the things we're very excited is, I was on the NIH Council. I was actually the one to help to put out the RFA on understand the microbiome metabolome. But in order to do that well, and in a repeatable condition, a well-controlled condition, you need those bacterial culture. And I think oral community will become one of the best ones because we have most of the bacteria already cultured. So you can really look at individual versus microbiome status and see how their metabolome changes.
Any more questions? Oh, one in the back. You can get a microphone back.
I'm sorry, between me and to lunch, so we can always catch up during the lunch.
I just have one query. Is there a tracking of the microbiome from newborn to adult?
Great question. So this is very much like the gut microbiome. The first 2 years was all over the place, but it kind of stabilized at the year or two. And then even though occasionally the international trip or antibiotics may disrupting in the short term, but in the long term, they very much go back to the same place. Although there is a kissing biology study showing, like in the college days, or when two couples gets together, that there will be some very frequent exchange, and the microbiome may merge together. But overall, they're very stable. But think about this: when you're getting a periodontal disease 60 years down the road, it was actually your grandma gave you when you're 2 years old. So that's why it's so important to actually have a saliva diagnostic when the woman gets pregnant and make sure seed the baby with the right microbiome. And that's something we try to do. Yes? So we would love to know more about it. I know with a lot of epidemiology studies and others and showing the connection. And so that the ear infections and nasal infections, this is the major concentration of the reservoir. And the lung and it is part of it. And we actually have some active study ongoing to finding certain bacteria like the Klebsiella and who is involved in the lung infection, and they could be also, they're a reservoir here. And so I do not have a particular answer to it, but this is the opportunity to call for more collaborations. And we're certainly hoping, to have a more opportunity to tagging on those kind of interesting questions.
I have a question.
Yeah.
So how does the food that we eat determine the microbes in the oral cavity, like the food choices that we make?
Yeah. So when you look at the archaeology data, our ancestor doesn't have much tooth decay until European to make the sugar canes. And I heard the story is why Elizabeth I never smiled is because she's the one can afford with all the sugar. She loves sugar. She has terrible teeth. And So there's certainly controlled nutrition has become something very important. And so prebiotic approach to modulating, oral microbiome is one of the very active approach. So Colgate is very actively trying to pushing out a new toothpaste, which is arginine based. So the concept is when you putting enough of the arginine into your oral cavity, you give the base-producing bacteria more upper hand so they can fighting off the acid-producing bacteria.
Thank you.
Thank you so much.
Sure.